Right, so this is going to be effectively part 2 of the fish room tour. Not because I've got a massive fish room, but basically because I forgot some of the stuff that I was going to talk about last time. So it's only a little bit. I didn't mention the overflow that I use on this tank, or the return lines that come back to it, and just the way I plumb them. So I'm going to do that first, and then I'm going to talk about something I put down over here, which is quoted a lot by various people in the hobby, and it is to do with the aerosolization of Amylidium mocellatum, which is marine velvet. So I want to go through the paper, which is the origin of the statement that people make about you can't have your quarantine tank in the same room as your display tank, and it has to be a minimum of 10 feet, so it's actually 3 meters away. So I'm going to come back and just run through that paper and actually have a look at the detail and see whether that statement holds any validity really. So I think first thing, I'll flip the camera around and just have a look at the overflow and just go through those returns that I was talking about. So bear with me. Okay, so there's the overflow. I think they call it a ghost weir or something like that because it's very shallow, and I can actually pull that off because it's connected just with 2 bulkheads to the box on the outside. So if I wanted to take that whole thing off and give it a good clean, which as you can see I don't do, I can do that. Maybe I will one day. Right, so this is the outside. So it's a black acrylic box. This overflow is made by Modular Marine, so it is American, and it does 300 gallons per hour. That's the maximum it can do, I'd imagine. Which is, I'm just trying to do the maths, is that 11,500 litres an hour, I think that translates to. The US gallons are slightly smaller than UK gallons. It's got 3 drains, which, because it's American, were 2-inch, so I converted to 50 mil, and I decided to fit 2 gate valves. One reason was just redundancy. Should one of the, so if I had one and it broke, obviously, then I'm out of luck. So if one of those gate valves does break, I mean, they're pretty good, don't really touch them that often, but should one break, I've got a backup. And the other reason I did it is, should I ever want to cut into my drain, you know, just say I wanted to put a frag tank under this tank, I could cut in, turn that valve completely shut, cut into that pipe and then drain it into the frag tank, and then replumb that frag tank into the sump. So it just gives me the option basically, that and to worry about having no flow from the tank if I want to work on one of those pipes. So that's it, and then obviously they all just go down into that amazing plumbing that I did that goes through the floor and into the sump below. So the only other thing I was going to talk about was the returns that come back up. So under the tank here, where are we? There we go. I plumbed in a pipe which connects that return and that return. So that return goes all the way around to the far end of the pump, and then this one comes up and basically goes to the end of that weir box that I just showed you. So that is shut at the moment. So I've got 2 return pumps on this tank. Each one runs one of these returns. So I figured if I ever want to service the pump, or should one of the pump breaks, which it shouldn't do because it's a biz pump, so it should last for years. But if I do have any problems, or if I just want to do cleaning or maintenance, I can turn that valve and then shut one of those pumps off, and then turn that single pump up so that I can get flow in the tank exactly the same as when I'm running 2 pumps, and then I haven't got to worry about rushing to try and do the maintenance, or if it breaks got to send it back to Germany, or whatever. So that was just one of those little things I put in. I thought it's pretty simple, but I've actually used it when I've cleaned my pumps. It works perfectly. So it just means I haven't got to turn the flow off in the pump, in the tank rather. Because to be honest, I never turn my flow off. I mean, the whole 2 years this tank's been running, I've never turned it off. So it's just not something I'm keen on doing, to be honest, because the ocean don't turn off. So, all right, what else is new? I have made a new screen for this end of the tank. Look, I've literally just fed these fish and they're all thinking they're going to get fed again. Yeah, so these are insect screens for windows that I buy off of eBay. That's a aluminium frame. I think it's powder coated. I buy the netting separately because it's white and a bigger gauge, whereas the netting you get is like a fly screen that you get with it, and it's really fine and black, so it would obscure the light. But all in all, they are about £30. It's lasted me 2 years. The only reason this one broke is because when I feed the fish, I have the food in one hand and I tend to lift this up with one hand like that, and it broke on that corner, obviously where the stress goes on the load. But the other three are all still good. So for £30 each, I got the whole tank cover for £120. Whereas when I looked at either custom made acrylic tops or the Red Sea or the D&D, because that's basically a 1 metre square, it's an unusual size for a tank to get a manufacturer to make a screen for, and they were just coming up ridiculously expensive. Right, so I think that's about it. I've got this actually, it came in the post today from Germany, and oh, can you see that? Not really. Actually, I'll take it off now. Yeah, so I've got this from Royal Exclusive. It is a new bracket for my pump on my skimmer. I've got an external skimmer, as you can see, and that kind of white bracket, you can't see it because it's on the other side, but over there, where that bolt is, I think it's that one. No, I think it's the bottom one actually. There's a crack through that stand, so I could have cracked it myself when I did the bolts up, to be honest, but I don't normally tighten things up that tight because there's an O-ring in there, so you don't really need to do them up that tight to get a seal. But anyway, so I noticed that cracked. I also noticed that the cable going into the pump had split, like this outer cable. So I don't know whether that's just because of the vibration on that. Doesn't really vibrate that much, but I mean, that pump's been a pain, to be honest, since I've had it, because it got sent out originally with the skimmer. It had no O-ring in it, so I set this whole thing up, plumbed it all in, and then water absolutely poured out the pump. And when I took it apart, the O-ring between this part wasn't there, and they forgot to fit it when they sent it out from Germany. And then also this little connection here weeped, so just get a slow drip come out, so I managed to super glue that to fix it. And then the pump was really noisy, so I took it apart again, and the ceramic block inside and the needle wheel were not properly fixed, and in the end it shattered and smashed to pieces, so they sent me a new one. So it's not been great, that pump. Hopefully I'll fit that new bracket and then I can just forget about it, because everyone tells me these pumps go for years and years and years, so that's what I'm hoping is going to happen. But so far it's less than 2 years old, and that's the list of problems I've had with it. Right, here's my observation tank with my tiny little black-tailed juvenile wrasse, my twisty eye wrasse, and my 2 grammars royal and black cap. So I do like that twisty eye wrasse, that's a nice fish, but he's been through the mill because he's been in my display tank twice now, and my 6 line does not like him. So at some point I think I'm going to have to take the 6 line wrasse out. Right, let me turn the camera back round. Whew. Right. So, right. So just not to do that really. Right. So that's the kind of end of the fish room tour. I think I've covered pretty much everything that's out here. So I was trying to think of what to do kind of long term for videos really. So it is a big tank and there probably will be stuff going on that I can make videos of. That's my oh, I can't show you that. That's my phytoplankton going in from the fridge, which is like up in the loft area on the ceiling part of this fish room. Yeah, so I've got what I was saying already. Oh yeah, long term, yeah, videos long term. I think it's very difficult to keep producing content other than doing tank updates, which I could do, but I find them a little bit repetitive, to be honest. So I thought I'm going to bring something to these videos which is probably an area that is interesting for me. I don't know if it's going to be interesting for most of the people to watch, but because of my background, so I have a PhD, I was a research scientist, academic for decades basically before I retired, and my area of expertise is in zoonotic diseases. And what I've kind of realised, particularly over the last 6 months, is that a lot of one of the drugs that are used in this hobby to treat fish and now corals as well are familiar to me because they're used in the medical world. So I did a master's at the London School of Hygiene and Top Med, which is basically a kind of medical school. I did a lot of parasitology as part of that masters, and so I thought, as I've got a background in science, I've lectured, I've taught, I've overseen master's projects, then I would look at the science which is ongoing at the moment and try and translate it into terms which are easy enough for the average hobbyist to kind of understand the basics of, and two, just to explain or to see how we can implement some of that science into our everyday reefing. So I'm going to start with, I might do like graphics and stuff or things as I get better, or not if it's too much work, but at the moment I'm going to just stand here and I'm going to explain a paper. So this is a paper. The reason I picked it is because it's quoted all the time, as I've already said, and so basically it is you can't have a quarantine tank within 10 foot of your display tank. So, and this is particularly, this paper is particularly dealing with marine velvet. So, where does that statement come from? Why is it that one line from that whole paper just been cherry picked and banded around? I don't really understand it, because I read the paper years ago, to be honest, and it always kind of makes me frown when people goes, oh, well, you've got to have a 10-foot distance between your quarantine tanks and your display tank. I mean, don't get me wrong, if you've got the opportunity to do that, then do it. If you've got the opportunity to have a quarantine tank in a different building than your display tank, perfect. But I think the crux of the issue is, ideally we want to get more people in this hobby quarantining their fish, and if you tell them that they can't do it in the same room or within 10 foot of their tank, there's a whole group of people which are going to go, well, I'm not going to do it at all then. And I think, well, I'm pretty sure that quarantining fish close to your display tank is infinitely better than not quarantining your fish at all. Even if you just do an observation tank before you plop them into your main display. So I'm going to go through this paper and look at what they did, how they did it, and just look at that one kind of thing that people keep quoting. So, marine velvet, I don't really know the life cycle. It's very similar to other parasites. Lots of parasites follow this same basic life cycle. There's no intermediate host, so it's literally the parasite and the fish. There's nothing else involved. So basically you get a trophon which attaches to the fish, the epithelium of the fish, which is just the skin of the fish, and the gills of the fish, which is why when you see fish shaking their head and flashing, that's because they got the irritation quite frequently in the gills. And then after a number of days that will fall off. I think it's 48 hours, but I'm not sure. It'll fall off, and then that becomes what they call a tromon. So that is basically a stage of replication. So you'll go from 1, and it will divide by cell division into, well, not millions, but quite often tens of thousands. And that sits in the substrate to do that. So it's not on the fish anymore, it's dropped off. It sits in the substrate, replicates, and then it kind of ruptures. Sporalization, I think, is one of the words we can use. And you get these things called dinosaurs. So this is the actual infectious stage. This is the stage which is going to go and hunt for fish to attach to, and then turn again into the trophon stage. So that's the kind of basic lifestyle. It's pretty simple. There's only 3 components to it. So what these researchers did, and they are based in University of Queensland, which is in Brisbane, is they wanted to prove that basically marine velvet can travel on very small air droplets through the air from an infected tank to a non-infected tank and infect the fish in that what was previously non-infected tank, because no one had done it. So what they did is they used juvenile barramundi, which is an Australian fish. They got infected fish, they gave them freshwater baths so that the trophons fell off, because freshwater makes them detach. That's why we do freshwater dips for our fish. And they put them through a nylon mesh of 47 microns, which traps them. And then they took them and put them in petri dishes with salt water at room temperature, and they waited for them to come into the dinospore stage, which is the infective stage. So they then put 100,000 dinospores into 2 litres of water, and they did what they call a static and a dynamic airflow scenario. So in the static one, they did first 3 trials by putting air stones into the infected tank, and then they had tanks set distance away from the infected tank. And I won't go into the details, but basically when they used an air stone in an infected tank, and the furthest away that those tanks were was a metre, so they were like 200 mil, 300 foot. Anyway, so the furthest ones, none of those tanks were infected. So with an air stone only, which does create some surface aerosolization, I would imagine, no tanks got infected. So they then changed it because they realised maybe we need to be a bit more aggressive about aerosolization. So they then put 100,000 of these dinospores into 2 litre garden mist sprayer. So none of the ones you pump. So they put 2 litres in there, pumped it up, and then they sprayed 100,000 of these dinospores in 2 minutes. So it's a litre a minute out of the spray. And even then, the only tanks that got infected were 440 millimetres away from that nozzle was the furthest. So anything further than that got no infections. And the tank under 440 mil, that's under half a metre, so it's a couple of foot basically, got infected. So they then did what they called a dynamic flow situation. So basically exactly the same scenario, but the tanks were spread out further. So they went from, I need to check back at the paper, 1 metre up to, it was 9 metres, was it? Not a lot, 5 metres, right. So 1 metre there's a tank, 2 metres a tank, 3 metres a tank, 4 metres a tank, 5 metres a tank. They used the same 2 litre sprayer, 100,000 dinospores in the water, And then what they did this time is they had a pedestal, 60 centimetre pedestal fan, 1 metre up in the air, and it was I think 60 centimetres behind the nozzle. So basically it's blowing an air current behind the nozzle, and then they released the 2 litres of water with all the spores in over 2 minutes, and it's blown with the fan, and then they looked at the infection rates in the tanks. So even then, even doing that, the tank that got infected the furthest distance away, which is 3 metres, which is I guess 10 foot, which is, I'd imagine, where this chuck away line comes from, about you got to add 10 metres. So that tank, so that they did 3 trials of that experiment, doing exactly the same thing 3 times. So you know, the replication is 3, and in one of the trials, one tank at 3 metres away got 1 trophon infection in it. So in 2 of the trials, the 3 metre tank got no infection at all. And that's, you got to remember, they're spraying it into the air with a fan, 100,000 dinospores in 2 minutes. So that infection is pretty low, I think. And if they'd done 2 trials, it could have been none. And they might have not had any infected. So all the numbers are pretty low. So they measured the trophons that came into the tank. Is it the trophons they measured? Yeah. Yeah. So that all tanks got less than 20. So this is a classic thing between the difference between our hobby and science. So that's science. The scientists are trying to prove that it is possible for marine velvet to survive aerosolization and infect another tank. And that's what they wanted to prove. And they had. They proved it. It does, and it did. That's a massive leap to go from that to say that in our hobby, in our environment, that we can't have a quarantine tank within 10 foot of your display tank. Because as far as I'm aware, you are not going to have an infected tank, take the water, spray it into your fish room in front of a fan and blow it all over the place. Now I know you have airflow in your fish room or in your house, but you don't have well, most of the time, you're not going to have a pedestal fan blowing at full speed, and you're not going to spray infected water into it. So I don't think it holds any merit, to be honest. I think you should be wary, but I think it's perfectly feasible to have a quarantine tank in the same room and reasonably close to your display tank. So the reason I'm saying that is because mine is. So my room is, I think it's 3 metres, its garage width. Is it 3 metres? It's about 3 metres, maybe slightly more. But anyway, it's way under 10 foot. So you can see my display tank behind me, and basically my quarantine tank is like 5 foot away from it. And I've quarantined every single fish in my tank. So that includes the 16 tangs, including the powder blue tang, the Achilles tang, which are 2, you know, quite prone icky fish. And as far as I'm aware, I've never seen any physical manifestations of disease, particularly, well, not velvet certainly, because that's quite lethal, or ick, to be honest. So I think that's a wrong statement to make. So that's just my take on it. So I thought I'd just do that video because when you actually read the paper, I don't think there's anyone in their right mind who's going to be spraying infected water into a fan where the display tank is, because that's just not the way we operate as hobbyists, is it? So that's the difference between how we operate as a hobbyist and how we try and prove something as a scientist. They're not always compatible. I mean, it's good to know, and it's good mentally to think you don't want to be splashing water all over the place, because if it does go up and in your tank, then you are taking a risk. But if you're careful and you take precautions, knowing that that's a possibility, then I really don't think that that statement's got any place in our quarantine for our hobby. Right, so that's I think that's basically it. can't think of anything else. So what else was I going to talk about? Yeah, so I think the other thing I was thinking about actually is that there is a lot of talk at the moment about antibiotics and disease in corals, basically. So brown jelly disease in torches and hammers, etc., and RTN and STN in stony SPS corals, stony corals. And I've noticed that a lot of the drugs which are being mentioned, so there's been a lot of talk recently about use of antibiotics. So things like erythromycin, which I think that's found in chemically, if I'm right, broad spectrum antibiotics. Other ones are like ciprofloxacin, kanamycin, things like this. They're all kind of broad spectrum. There's another one as well, oxalic acid, I think is the other one, which I actually think oxalic acid will probably be better for reefers in the UK because you can buy that as a powder. It's a synthetic antibiotic, and as far as I'm aware, it's not what they call a POM, which is a prescription only medication. So I think you can get that off of like APC chemicals. It's not as strong, I don't think, as ciprofloxacin, but it could work as well. So the point I'm trying to make it is a lot of these diseases, which, well, not a lot of them, but some of the times when you've got RTN and STN in your tank, it is because of a bacterial infection. I mean, there's a myriad other reasons which could cause the same thing, but I know people are just starting to chuck antibiotics in their tank, and they say they're having good results. I think that's just a dangerous precedent to start setting. What I would actually like to see is those people which are at that sort of cutting edge of using these antibiotics, and I don't know what it is because I'm in the States, so I don't know. I'm in the States? I'm not in the States, I'm in the UK. We have quite strict rules on how we can obtain antibiotics, and I don't think those restrictions apply in the US. I just get the feeling that they're able to get their antibiotics a lot easier, it seems, without the need of a pharmacy or a doctor making the prescription. I could be wrong, but I kind of get that feeling. Which is why I'm saying I think that oxalic acid might be a better bet for the UK people, because over here you're going to have to try and get a pharmacist to give you ciprofloxacin, because we use it, well, it treats diarrhea basically. You've got bad diarrhea, it's one of the medications you can take for that. So what I was trying to get to was, because these are broad spectrum antibiotics used in the medical profession for humans, and as most of us know, there's a big problem of antibiotic resistance, I would like to see those people, particularly those people in the States which are spearheading this kind of approach to treating STN and RTN using antibiotics, is just as important as treating the coral is how you treat the waste water from those dips or whole tank treatments. So you need to find a way to prove that it either breaks down naturally in our tanks with either the use of UV, carbon or ozone, or a mechanism which people that want to use it can employ, so that when you tip it down the drain you're not going to be participating in antibiotic resistance globally because you wanted to save your torch coral. So that's kind of where I am. I like the science, I understand it's exciting, especially in the States where you can do, what's that company, Bionomics is it, where you can send your water sample off and they will give you a complete breakdown of your bacterial load in your tank, the diversity of it. And it would appear from what I've read so far that two main causes of I think this is an SPS, is Vibrio bacteria. I mean, the famous Vibrio bacteria, which I think probably everyone in the world knows, is cholera. That's a Vibrio. Vibrio cholera is the organism which causes cholera globally. So that's a nasty disease. So you should remember this when you're getting a mouthful of tank water as well. It's not all nice that's in there. And then the other ones are, is it Archobacter, which are, I think they're probably to a lesser extent, but they seem to be potentially causing a problem as well. And interestingly, now this is a little weird thing, because I always microwave my frozen food, always have, because, and this is just the way I think, I guess. So companies that make fish food that is not live food, they don't, I could be doing them a massive disservice, but this is just the way I think. This is not, there's no evidence to this, you know, this is just the way I'm thinking. Companies that produce fish food, like frozen mices, you know, reef feast, all the things that I feed, the brine shrimp, all of this, when they're live cultures, obviously they've got to be reasonably sterile and clean, otherwise you get a culture crash. But when they're frozen and when they're packaged before they're frozen, I don't know how good their business model needs to be in terms of making that food. I mean, I know they say it's gamma irradiated, isn't it, some of it, but the process after that and the fact that it travels some distance, it could be even globally, I guess, it could thaw, re-thaw. I don't think there's like a perfect cold chain for fish food like there is for human food even. And so because of that, I was always wary of what was in it, in terms of anything. It might be, one, it might be off, you know, am I feeding my fish food which is thawed out 2 weeks ago, sat in the sun, and then it's been refrozen and sent to my local LFS, and now I'm going to thaw it out and feed it to my fish. So I've always had those kind of niggling doubts about the food. So one of the ways that I thought I would deal with anything that's potentially harmful is I always microwave my food I actually microwave it for 30 seconds on full power. So in my kind of eyes, that would kill any harmful bacteria. Because the reason I'm saying this, I think, I can't remember what it is, it might be letter actually. He's saying that Archobacter can survive in frozen food. So I haven't actually seen any papers that have actually said that's true. I've had a look. It does say that they've got some of the same genotypes for cold tolerance that Campylobacter have got, which is another type of bacteria. So, but that only went down to 8 degrees. But it is quite possible. So yeah, so that's just a little snippet. So if you feed frozen food, don't leave it on the side to thaw out. Stick it in the microwave for a minimum of 20 seconds. Yeah, it's just one of those little things. I just thought it was weird because I've always done it. So I'm not going to say it's not because that's the reason I've never had STM, but it's just one of those things. This is easy to do, and so if potentially it can save you from getting some harmful Archobacteria in your tank, then it's worth doing. Right, so I think that's about it. Yeah, so going forward, I don't know, I said this already, I've talked for so long I've forgotten what I've said. Yeah, so going forward, obviously, because I've got a scientific background and I find these things interesting, I will probably just present little bits like this, papers, you know, try and make sense of it, see whether it's applicable to our hobby or not. So I'll do a quick tour of the tank when I do these videos. And oh, I know what I was going to say, I knew there was something else. I was talking to Paul at AAC the other day about the way that I quarantine my fish. So I use a hybrid tank transfer method for, I'm going to say, all of my fish. I was doing it for all of my fish until I got to the wrasse, and then they're just a pain in the arse basically. But I probably will still do it, I'm just slightly changing it. But essentially for everything else, and I put a clean wrasse, a 6-time wrasse, an iridis wrasse and a yellowtail wrasse through this hybrid tank transfer method. So it's something I've invented on the shoulders of giants. It's a classic. So I think it's Humble Fish and his Borum which have kind of paved the way for this hydrogen peroxide and tank transfer. So tank transfer is just every 72 hours you are changing your fish out to a new sterile tank. And the reason it's called hybrid is at the beginning you do a hydrogen peroxide bath, and then 6 days, so after you've done 2 transfers, because they're basically 3 days each, you do another hydrogen peroxide bath. And then I also do 2 treatments with API General Cure, which is basically praziquantel and metronidazole? I think it is, can't remember off the top of my head. Yeah, so basically for intestinal worms and flukes, etc. Yeah, so I've got some fish over there. I've got a black cat grammer and a royal grammer, which I'm going to put through probably in the next couple of weeks. So I'll make a video of all the steps that I do. So yeah, so that'll be a future video as well. So I've been chatting for ages now. Yeah, so that's it. So if you're interested in any of that, you can look at the tank update, which I'll probably do, and then you can just turn off if the rest of it don't interest you. And then I'll probably do like a little science segment of what I find interesting at that particular moment. And then I'll do something like the my QT system, or I'll talk about the UVs I run, or I'll talk about how the ozone has worked, how it works on my tank. Yeah, I mean, actually ozone I think is going to be quite interesting in the future, because if you run ozone at a high enough level, you will kill bacteria. So most of these bacteria which seem to be causing disease are gram-negative bacteria. So you get gram-negative, gram-positive. It's just the way they stain. Basically, it's to do with the membrane that they have, whether it takes up a dye when they stain them or not. Yeah. So yeah, I think that's it really. So yeah, if any of that interests you, you can either watch all the videos or just pick the bit you want. I'm sure if I get feedback which is reasonably positive, I will probably put more effort into, you know, making the videos a bit more snazzy. But yeah, that's basically where I'm at the moment. So thanks for watching. That's been quite long actually. And I will see you. Like, one last thing, I'll probably make 1 a month. I reckon that's, if I say I'm going to make 2 a month, 1, I'll put myself under pressure, and 2, I'll probably make myself out to be a liar. So I reckon 1 a month will probably do it. I might do very short ones, because I post on Instagram like 3 or 4 times a week, but they're just like little short videos, no effort. So these YouTube videos, editing, blah blah blah, it's just they just take quite a long time really. So I reckon 1 of these a month will probably be my limit. So thanks for watching, and I will see you in the next one.